హలో భవాని మేడం ఇచ్చేయండి, భవానీ గారికి ఇచ్చేయండి. కనకరాజ్ సార్ కూడా ఉంటారు చూడండి. హలో 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 హలో
Hello.
Hello? 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 Kondababu garu, is it audible to you? Kondababu garu. కొండబాబు గారు కొండబాబు గారు వినబడుతుందా నా మాట? కొండబాబు గారు
Hello.
హలో కొండబాబు గారు.
Hello.
కొండబాబు గారు, is it audible to you? Hello?
Can you hear me?
Yes, but somewhat disturbance in your voice. Reduce the echo, sir. Reduce the echo. Kondababu garu.
Is it okay? Is it okay now?
It is okay, sir. Is it okay?
Thank you.
Let us start. 2 more minutes are there. So shall I exit my screen, sir?
Yes.
Just put your slide, first slide onto the screen. Just I am offing my screen share. Okay.
Yeah. What do you like to share? Is it possible to see my screen?
Yes, sir. Yes.
Okay. So there's 1 minute. Maybe perhaps I will wait.
Yes, sir. Okay. A very great pleasure to meet you all again and again. Let us use this lockdown period effectively to gain and enrich our knowledge. I am Dr. Fanikumar, Principal, Adarsa College of Pharmacy. On behalf of Adarsa College of Pharmacy and Koringa College of Pharmacy, would like to say you a grand welcome to this international webinar session. A very good morning to beloved Chairman, Adarsa Educational Society, Sri P. Kanakaraj Garu, and all directors, Koringa College of Pharmacy, Secretary, Adarsa Educational Society, Sri P. Nagmani Garu, Coordinator, Adarsa College of Pharmacy, Sri P. Srinivas Garu, AO, Adarsa College of Pharmacy, Sri Sivaramakrishna Garu, Principal, Koringa College of Pharmacy, Dr. Bhavani, and all participants. And last but not least, to today's speaker of the webinar, Dr. Konda. Sorry for the disturbance. Last but not least, today's speaker of the webinar, Dr. Kondababu Korakla, Associate Professor, Leiden University of Medical Center, Netherlands. First, let us start this session with the introduction of Dr. Kondababu Korakla. Now I ask Dr. Bhaskar, Associate Professor, Adarsa College.
Thank you, sir. Thank you, sir.
Hello?
Thank you. Hello.
Yeah.
Is it audible, sir?
Yeah.
Okay. Thank you, sir. A very good morning to today's speaker, Dr. Kondababu Garu and Sri Kanakara. And Sri Kanakaraj Garu, Director, Adarsa College of Pharmacy, and Sri Nagamani Garu, Secretary, Adarsa College of Pharmacy, Sri Srinivas Garu, Adarsa College of Pharmacy Coordinator, and Sri Shivaram Garu, Administrative Officer, Adarsa College of Pharmacy, and Dr. CHS Panikumar Garu, Principal, Adarsa College of Pharmacy, and Dr. Bhavani Garu, Principal, Koringa College of Pharmacy, and everyone who are participating in this webinar. I'm glad to introduce about Dr. Kondababu Kurakula. He has completed his D.Pharmacy from Government Polytechnic, Visakhapatnam, and B.Pharmacy from Vishnu Bharati College of Pharmacy, Guntur. And he has completed his MS in Molecular Biology at University of Skövde, Sweden. He did PhD from Amsterdam University, Netherlands, postdoctoral fellowship at LUMC, Leiden University Medical Center, Netherlands. Presently, he is working as associate professor at Leiden University Medical Center, Netherlands. He has awarded from the various societies with more than 15 awards. He had 32 publications at international level with average impact factor 8. He was attended 18 international meetings also. He did the certification, namely animal handling, biostatistics, and vascular biology courses only. If I missed out any of your profile, it is out of my reach or lack of information. Thank you, sir. Over to Dr. Fani, sir.
Thank you, Bhaskar, Dr. Bhaskar, for a good introduction regarding today's speaker, Dr. Kondababu Garu Korakla. A great profile at his young age. I hope all will grasp all these things from him, and I hope all you will reach to the same level. Thank you. Now I am handovering the session to the Kondababu Garu. Please, Kondababu Garu, carry out the session, please.
Okay, thank you all. Thank you, Dr. Fani and Dr. Bhaskar, and also the rest of the management and the staff from the Adarsa College of Pharmacy as well as from the Koringa College of Pharmacy. I hope everybody can hear me well. So I assume it's you can hear me. So in this next 30 minutes or so, I would like to give a brief overview of the research what's going on in this disease called pulmonary arterial hypertension. So this presentation actually involves multidisciplinary subjects. For example, it involves pharmacy. It also involves molecular cell biology. It also involves chemistry as well as clinical medicine. So I try to make it as simple as possible to make sure everybody can understand this presentation. Hello, can you hear me? I think this is not moving. Yeah.
Yes, sir.
Okay. So in the normal lungs, as you can see, I cannot show my cursor. So in the left side, you can see that normally we have the blood vessels in the lungs, like every organ in the body, and we need the blood vessels, of course, in the lungs to get the gas exchange. And the vessels in these lungs, in normal human beings, actually are widely open for the efficient transfer of the nutrients and everything. However, in the disease scenario like pulmonary hypertension, these blood vessels, which is pointed out in the arrow, can be constricted. That means there is because of abnormal remodeling is going on. So the blood vessels in these lungs of these patients are completely occluded. So as you can see in this schematic below. So because of this abnormal constriction of the blood vessels, there is increased pressure in the right ventricle because the right ventricle has to work very hard to pump more blood for the exchange of the gases. However, because the abnormal remodeling that is present in these patients can actually lead to increased pressure in the right ventricle, and eventually the right ventricle is collapsing. So the survival of this patient is very limited. So this abnormal remodeling happens in these patients actually involves multiple processes, such as cell growth is being increased, because normally if you have one layer, in these patients you have multiple layers. There is inflammation both locally as well as systemically in these patients. The metabolism of these cells present in these lungs of the patients also substantially being increased. And there are also increasing of the different signaling pathways such as TGF-beta signaling, as well as there is a downregulation of BMP signaling happening. So a lot of things are happening in these patients, and as you can see in this screen in the left side, the vessels in the lung are completely occluded. So this is a specimen from histology specimen from a patient who actually been died. So as you can see, the blood vessel is completely narrowed. That means the lumen is completely narrowed. There is more proliferation of the cells. There is almost no lumen left for the patient to get for efficient gas exchange. This phenomenon is can be seen also in this overview of the lungs. So on the top side of this, you can see the healthy lungs. On the lower side, you can see the lungs from the PH patients. So all the in the gas to be swapped. So the top one is pH and the lower one is healthy. So in the healthy vessel, in the healthy patients, you can see the vessels are completely open, whereas in the patient's scenario, you can see that the black dots actually represent the vascular remodeling. So same phenomenon that you can see in almost all pH patients. So the vessels are completely occluded in at least 40 to 50% of the patients, patient lungs. That's even if occlusion is 30 to 40% is enough to kill the patients. So this is very serious complication. So we call it as a deadly disease because of this phenomenon. This abnormal remodeling of the vessels actually taking place in almost all the vessels, but there is a different grading. So not all the vessels are completely occluded. So there is a grading called grade 1, grade 2, grade 3, and grade 4. Basically, what it represents is the more grade, the more remodeling. That means more cells are being occluding the vessels in the lungs of these patients. The patients either can die when the grading is 2 to 4. Because of this grading is increasing, so the abnormal remodeling is increasing, there is increased pressure in the pulmonary artery. The pulmonary artery so will be constricted. So the pressures in the pulmonary artery is been substantially being increased. So that means there is increased pressure in these patients. So PH survival is very a lot, and it varies among the different ethnicities and races. From one of the registries from the US, it can be said that the average survival is very limited. And so on average, the patients die if they are not treated. They die between 2 to 3 years. So that means this is very serious complication compared to many cancers as well. Fortunately, this disease is very rare. But if they, on the other hand, we assume that the patients die because of the right ventricle failure, but people assume that in the clinic or also outside, they died because of the left ventricle failure. So that means patient maybe not completely being diagnosed as PAH, but they die. So the survival is very, very poor, and the treatment that is available so far is very limited. So the treatment is only working on the symptoms rather than on the actual root cause. So that currently there is no real treatment available to actually reverse these underlying processes in this lung tissue of these patients. Another important aspect in the disease of PAH is females. Females are highly likely to get this disease much stronger than male. And we don't know exactly why, but at least depending on the type of the disease, at least females are at least can get 4 times to 9 times to get the disease. So there is something going on either with the estrogen hormones or with the X chromosome that is present in the females compared to the men. But this is currently under investigation by our team. And although the problem lies in the lungs, the patients die because of the right ventricle. So the right ventricle of the patients actually collapse. As you can see in this left side of the screen, the right ventricle of these patients actually becoming enlarged in the red color. And this right ventricle becomes eventually become thin, and eventually it fails. So this process can take a couple of years, and then the patient that is presented on the right side is actually the ones we see in the clinic. So the patient comes to the clinic very late. To give you this example of why this patient comes to the clinic is because of the lack of oxygen. So they actually cannot breathe that well. The patients are tired because there's less oxygen can be taken. And some serious patients can also even cannot tie up their shoes. So that means this is a very serious complication. There are different kind of PAH present. So PAH can be because of genetic mutations that only counts for only 4% of the patients. Majority of the patients doesn't have any mutations known. Thus we call them as idiopathic PAH patients. There are also patients with HIV infection, for example. Some drugs which are being used in the cancer, also in other diseases, can also induce PAH. And some diseases like congenital heart disease, patients with two cuspids or instead of three cuspids can also have this disease. Some patients with the liver disease also can get this disease. The patient which has any of these complications die faster than the patient without any mutations or any other cause known. So basically, the patient without known mutation, like idiopathic patients, die lesser than the patient with HIV or drugs or with the liver disease or other liver disease and connective heart disease. So the problem in the abnormal remodeling is there are many factors involved. So some people assume that the pathology of PAH patients in lung tissue resembles more like cancer. So in cancer, we know most of the cases, there is excessive proliferation of the cells. The cells grow very faster than they should. There is also a lot of stress, oxidative stress, for example. There is also inflammation problem going on. There are a lot of things going on in cancer. Such same characteristics also going in this disease as well. So we know there are some genetic factors known. There is inflammation is going on in these patients. So on the top side, you can see the healthy tissue in the lung vessel. So typically you have only 3 layers of the cells: endothelial cells, smooth muscle cells, and fibroblasts. They work together in every blood vessel of the body, including the lungs, to make sure the vessels are completely constricted or properly constricted or dilated, depending on the necessity to maintain the homeostasis of the vessel. However, in the disease scenario like pulmonary hypertension, instead of having 1 layer or 2 layer of cells, the cells proliferate too many. So that means the vessels are being occluded, the lumen is becoming narrowed, and that means the blood cannot flow through these blood vessels in the lungs. So they cannot take the blood, and the right ventricle fails. So there's some, this phenomenon is going on. There is a proliferation, I've told you, and there is increased inflammation is going on, and metabolism is increased, and there is a signaling called TGP signaling going on. I'll briefly explain this signaling in a minute. At the same time, in this same lung tissue, there is a BMP signaling is also going down. The classification of pH actually is at different forms, like I mentioned before. Some of the mutations known in this disease are linked to some genes that are highlighted here in the yellow color. So BMPR2, ALK1, endoclin, SMAD9, caveolin-1, they actually all belongs to a family called TGBeta BMP signaling pathway. So in pharmacology, we only talk about the at the surface receptors most of the cases. For example, like GPCRs and so on. In the molecular biology and in the clinical medicine, we also discuss much more into the signaling as well. What's going in the cell? It's very important to understand the function of a gene or a transcription factor or a drug. So I will try to explain this schematic very in a most easiest possible way. So these receptors of the BMP and TGBeta are actually present on the surface of the cell. So they're exposed to the blood vessel in these endothelial cells of the patients. For example, the ligands like BMP or TGBeta bind to their respective receptors. They actually lead to different signaling pathways. It's unfortunate that I cannot use my arrow to show you. So the BMP ligands bind to the receptors like BMPR2. They actually increase the signaling called as phospho-SMAD158. To get this SMAD158, SMAD4 goes to the nucleus of the cell. In the nucleus, we have the DNA, and in the DNA, they bind to their respective target genes. And these target genes, such as ID1, ID3, that are presented here, actually can mediate the cell function. The fate of the cell can be determined by this signaling pathway. So, for example, BMP ligands actually present in our blood, or TGBeta ligands present in our blood of the patients, are also in normal human beings. So they actually being secreted, and they are also being taken up the cells, neighboring cells. So these ligands actually bind to the receptors. They go to the nucleus of the cells, and the nucleus they bind to the DNA of specific target genes. These specific target genes can actually mediate the fate of the cell. So in the disease like PAH, there are some mutations known in this pathway. For example, BMPR2 pathway is mutated, and this ALK1 is mutated, endoclin is mutated, SMAD4 is mutated. So that means the DNA of this in these specific genes has been mutated in different locations. So that means this signaling pathway in these patients is being disturbed. So instead of having, let's say, 1 unit of ID1, it's having maybe 2 units or 3 units or so on. So the pathway is completely disturbed in these patients. That may be the reason why the patients are dying. So in a nutshell, so what's happening in these patients, the BMP pathway is being disturbed. So it's being reduced. At the same time, the neighboring pathway, TGBA pathway, is being increased in these patients. So this is everything happening in a cell of a tissue in the body of these patients. So if you take the serum plasma from these patients, for example, what you can see that if you look at the levels of TGBeta1 by using an ELISA to look at the TGBeta protein levels, you can see that the levels of TGBeta is substantially being increased in these patients. So we have 2 different groups of patients here. IPH, idiopathic patients, there is no mutation known. In the hereditary PH patients, there is mutations known. What you can see in both cases, independent of the mutation, you can see the levels of TGBeta levels are going up. So that actually basically suggesting that mutation doesn't matter. The levels in these patients are substantially being increased in this process. In the same patients, there is a mutation known, there is BMPR2 mutations known. Again, these mutations are present in the DNA of these patients. So the BMPR2 mutations are present in these patients. Unfortunately, the mutations that we know so far in these patients are more than 350. So that means the mutations are present throughout the DNA of this BMPR2 gene present in these patients. So it's impossible to correct these mutations using novel technologies like CRISPR-Cas and traditional gene therapy. So if there is one mutation, for example, known, perhaps we could have tried gene therapy to correct this mutation and to correct this mutation to save these patients. However, it's very unlikely to correct these all 350 mutations, and these mutations are different between different countries, different races, different ethnicities. So that means it's impossible, almost impossible, to create a gene therapy for all these patients because the mutations are very different. For example, like the COVID-19. For example, we know that now in India we have 198 COVID-19 mutations known. So that means it's highly unlikely to use a gene therapy or something to correct these mutations to save the COVID-19 patients. The same scenario also applies to this BMPR2. The mutations are in this case happening in the DNA, whereas in COVID-19, of course, it's in the RNA. So the gene therapy is not a solution. So we're actually looking for solutions which can actually make this DNA, the signaling of the BMPR signaling is can be changed by using different means. So we use a pharmacology approach, we use a genetic approach, we use a combination approach as well to understand. So in a disease scenario, in the left side, you can see that in the different cells, these receptors, these ligands bind to different receptors in the cell of the tissue. And in the disease scenario like a PAH, this in the black dot, you can see that mutations actually lead to the receptor is not functional. So if the receptor is not functional, you cannot change the signaling which is happening in the cell of the tissue. So therefore, we wanted to use different approaches to maintain this balance. How
ఈ ఆడియో మ్యూట్ అయినట్టు కొంచెం చూడండి సార్. అనుభవ్ సార్, ఈ ఆడియో మ్యూట్ అయినట్టు కొంచెం చూడండి. ఇది నాట్ ఆడిబుల్ టు మీ.
Okay. It looks okay for me. Can you hear me now? Hello? Hello, can you hear me?
Okay, sir. Okay, sir.
Is it okay now?
Okay, sir.
Okay. Yeah, I think. I think there's some delay going on. Probably some networking issues. I can't even move. Something is going wrong. Can you see me now? Can you hear me? Can one of you can confirm?
Yeah. Yes, sir.
Okay.
You can carry on.
Yeah. Sorry for the disturbance. Yeah. Okay. So there's a problem in the restoring the balance between the TG-beta BMP signaling. So this can also be detected here. So there is a problem of this receptor mutation in the BMPA2, for example. There is a problem of TG-beta receptor. So the TG-beta signaling is being increased, whereas the BMP signaling is decreased in these patients. So therefore, what we are trying to aim what we means the whole pH research field across the globe is trying to rescue this balance between the TG beta and BMP. So by maintaining this balance, we assume that we can repair the lungs, thereby we can save the heart of these patients. So there are different approaches that has been proposed to be used to maintain this balance between this TG beta and BMP. One of the approaches is to use a ligand which actually binds to this BMP2 receptor. So one of the ligands which has been known is called BMP ligand, which is also depicted in this right side of the screen. The BMP ligand actually binds to BMP2. So so it seems like the BMP2 expression is substantially low in the lungs of these patients, and importantly, patients who have a mutation of BMP2 or no mutation, all the patients have a problem in the BMP signaling. So BMP signaling is low, independent of the mutation in all the patients. So we also in my presentation, I'm going to talk about 4 different compounds which we actually worked on, and I also would like to emphasize the success of these compounds so far in the PH community to the patients. So I'm going to talk about BMP ligand, which actually binds to BMP2. I'm also going to talk about FK506, which is also known as the tacrolimus, which is being used in the drug-eluting stents, for example, which actually binds to a protein called FKBP12. I'm also going to briefly discuss about the results from the 6-MP, 6-mercaptopurine, also known as azathioprine, which is being used in the clinic over 60 years or 70 years and it's also gold standard drug for the inflammatory bowel disease or Crohn's disease. And this 6-mercaptopurine actually targets a gene called NUR77. And most recently, we developed a drug called RVX208, and this drug also works on a different signaling pathway in this whole cascade. I won't go to details in this about signaling pathways because of the audience, but I will briefly explain how these drugs are actually working on different aspects of the disease. So first thing is we want to understand how this BMP9 ligand it works. BMPs are proteins are being circulated in the body of every human individual. So healthy individuals as well as patients have BMP9. We think the patient's pH doesn't have enough BMP9. That's why there is a decreased signaling, and that's why we think that patient is developing a disease compared to the normal people. So since BMP9 levels are low in these patients, we gave BMP9 protein, which is made in the lab, to these rats. To give an example, what we did first is we took the lungs, for example, from the patients who died, and we took the cells, we isolated cells from these lungs from these patients, which is in the middle of the vessels that are occluded. We isolated cells, which you can see in this bright field image. So each this bright field image shows cells isolated from these PH patients. So for these patients cells, which is cultured in the lab, we actually gave BMP9. We try to look at different signaling pathway, which is problematic in these patients. This is we took control, which is basically nothing in the middle screen, which is a Western blot, to detect the amount of protein. So we use BMP9 to this, and we gave BMP9 to the cells, and we looked at the downstream signaling of this signaling pathway. So we look at SMAD158 phosphorylation. As you can see on the left side, in the middle Western blotting, you can see that BMP9 can substantially increase phospho-SMAD158 levels, which is also seen on the downstream genes which is present in the cells. So normally, in the normal individuals, the signaling pathway is not changed, which is normal, but in the PH patient, the signaling pathway is downregulated. So that's why we want to give BMP9 to increase the signaling pathway in these patients. So we tested this by using number of different approaches. For example, in this case, I'm showing only quantitative PCR as well as Western blotting to look at the levels in these patients. So everything that we did, we went from a patient approach. So we went for the patient cells, patient tissue, patient samples directly. to validate to find our findings, and then we went back one step back, and we went back to the different cells and different animal models to validate our findings, and then we went back to the clinic again. So it's a different approach than traditional approach. So since the finding that we have in the in vitro cell culture are very promising, what we did is we tested this BMP9 by giving to some rats which already developed pH. So we use a different model called Susan hypoxia rat model. I won't go to details, but what you see in this model, the vessels in these rats in the lungs actually mimic the same as in the patients. The vessels are completely occluded in these rats. Once they are occluded, we started giving by BMP9 or vehicle group. So in this case, we gave phosphate buffer saline, PBS, as a control. So we gave BMP or we gave just PBS. In this slide, you can appreciate that the BMP9 can actually reverse the remodeling that you can see. So the lower panel, you can see the BMP9 treatment animals, and the upper panel, you can see the saline PBS treated animals. So the vessels, as you can see, this is one micro vessel. So this micro vessels actually kind of completely occluded. There is no place for the blood to flow like you see in the patients. And when you give BMP9, as you can appreciate that the vessels are completely open. So this actually shows that BMP9 treatment is actually working in the lungs of these patients, the lungs of these rats. We also look at number of what it has been known that some drugs only works in the lungs and they don't repair the heart. So this is a problem because the patient died because of the right ventricle failure, and the patient has a problem in the lungs. So you need to repair the lungs to improve the right ventricle. But sometimes it happens that the right ventricle is not repaired, whereas the lungs are repaired. So that's not a good solution. So we always look for the lungs and the heart as well. In this case, I'm showing some data on the right ventricle systolic pressure, which is normally very high in these patients. So in this case, I'm talking about, of course, rats. So when as you can appreciate that giving PMP9 is substantially decreasing the right ventricle pressure as well as the right ventricle size. So right ventricle hypertrophy is being decreased. The size as well as the pressures are being decreased in the right ventricle, and the same also applies in the lungs of these rats. So both lungs and the right ventricle are improved by giving BMP9 treatment. One of the biggest problem with BMPs is they can actually induce bones. So that means calcification. So BMP stands for bone morphogenetic protein, B for bone and M for morpho, and P for protein. So these bone morphogenetic proteins are actually known to induce bones in the body. So that's why BMPs are very important in the embryonic development as well as in the late stage development for our skeletal muscle development throughout the body. So what we try to do is our collaborator actually tried to do is they gave the BMP9, but actually it's repairing both the lungs and the heart, but actually it has some implications to induce some calcification. So we want to repair the lungs and the heart, but we don't want to induce any side effects. So this is an example of alizarin red staining, which is in the red color on the right side. And we use different endothelial cells. We give BMP6 or BMP9 in some cases. What you can see, the more red color means more bone formation. So what is happening on the long run, if you give the BMP9 to the rats, they actually develop bones in the lung or in the right ventricle or in other parts of the body unnecessarily, which is a very serious side effects. Therefore, so continuation of BMP9 is not a good solution. So perhaps if we can deliver the BMP9 locally to the lungs, that might be beneficial compared to the giving BMP9 systemically to these rats or to the patients. Currently, there is one startup company has been started with this BMP9 treatment to make this trial, these findings really true in the patients. That's from the Oxford and Imperial College London in the UK. So since BMP9 is not so promising, although it's working the way it should, so we actually started looking for different approaches. So what we did is we used a drug repurposing purpose. we did a big screening using more than 33,000 compounds. All of them are FDA approved compounds, and we used BMP signaling as a readout. And in this high throughput screening, we identified FK506 or tacrolimus as one of the compounds which actually increases the signaling of the BMPR2. So I'm going to discuss one or two slides on FK506. So FK506 is increasing the BMP signaling and inhibiting the TG beta signaling. FK506 protein actually binds to this specific protein on the FKBP12. This FKBP12 is a protein which actually binds to BMPR2 receptor. So BMPR2 receptor is present on the surface of every cell of the human body, but FKBP12 is present only inside the cell, and is actually binding to the BMPR2 receptor indirectly, and thereby stopping the BMPR2 receptor not to function in these patients. So the more FKBP12 expression is present in these patients, therefore, by using FK506 compound, we want to stop the FKBP12 expression. Therefore, we want to save the patients. So we used, again, Western blotting and different approaches to identify in these patient samples. And most importantly, FK506 stands here FK. The FK506 can increase the PMP signaling, which is a readout from the phosphosmad 158 or ID1. The expression of phosphosmad 158 or ID1 expression is substantially being increased if you give FK506 only for 1 hour. So 1 hour is already enough to increase the signaling, which is impaired in these patients. So FK506 is increasing the BMP signaling in these patients. Again, we use the same animal model as before. We gave FK506 our vehicle group to treat these animals which actually have this disease. So, and we can see that FK506 can actually stop the progression of the remodeling. So if you compare the vessels in the vehicle versus FK, the vessel is completely open. The white space indicates actually the lumen is open. That means the blood can flow easily in these rats. Whereas in the vehicle group, the vessel is completely occluded. So there is no place for the blood to flow. That's why these rats die if you leave them for some time, like the patients. So since the data is very promising from FK506, we started as a proof of concept clinical trial in the patients. So we started a patient who is a female. This is done at Stanford University in the US with one of our collaborators. As you can see that FK506 can actually improve the patient symptoms substantially. At the same time, the patient is still alive. This is more than 40 months now. Typically, the patient die from 1 to 2 to 3 years. So if the patient is still alive, the FK506 is working very well in these patients. However, there are some side effects that should be considered. So there's a large clinical trial is being planned, and currently we are waiting for the funding to be obtained to continue this clinical trial. And next to that, we also interested in looking for different molecules. We used a molecule called 6-mercaptopurine. 6-mercaptopurine targets NUR77, a gene called which is present in this lung tissue as well. And we found that NUR77 is actually it modulates both BMP signaling and TGP signaling. So therefore, we actually look for the expression of this NUR77 in the lung tissue from these patients. And what you can see on the left side, in the lung tissue of these patients, the expression of NUR77 is substantially being decreased. And the same also applies in the lung tissue, in the endothelial cells isolated from these patients as well. To give an example of how these cells look like in this lung tissue, you can see that in the right side, the cells from the control individuals and as well as from the PH individuals. So if you take the cells from this patient and if you put them outside in the plastic, in a culture dish, you can see that the patient cells proliferate very fast. So even within 14 hours, the cells proliferate 2 times compared to the control individuals. So the expression in this of NUR77 gene present in this patient is substantially being decreased, and therefore we want to increase the NUR77 expression to stop the progression of this disease. So what we did is we gave the 6-mercaptopurine, which is also known as a prodrug of azathioprine. We gave 6-mercaptopurine to these animals, and what we can see in these vessels, which is also BMPR2, can be seen in the green color. The BMPR2 expression is substantially being increased after giving 6MP compared to the vehicle group. So the same also can be seen on the right side. 6MP can increase the BMPR2 expression levels as well as the downstream signaling. So 6-mercaptopurine is reaching the lung tissue, although we gave away the 6-mercaptopurine by drinking water, and as you can see that 6-mercaptopurine also can reduce the remodeling happening in this patient. So the 6-mercaptopurine is decreasing expression of increasing the expression of BMPR2. At the same time, it's also decreasing the vascular remodeling that is present in this rat. It's not only that, 6-mercaptopurine is improving the cardiac function. So it's improving the right ventricle function as well. The cardiac output, which is normally decreased, the patients die because there is less cardiac output. So in this case, when you're giving 6-mercaptopurine, it can actually increase the cardiac output. The stroke volume is also being increased. So 6-mercaptopurine is doing what it should in these rats. It's also doing a lot of other stuff in the right ventricle of these patients. So in the on the left side in the F, you can see that the cross-section area of these patients is of these rats is being decreased. So typically, the cardiomyocyte size become enlarged in these rats. But when you're giving 6-mercaptopurine, you can actually see the cardiomyocyte size is being decreased in these rats. And the same also applies for the fibrosis. The fibrosis staining on this right ventricle tissue from these rats can also be decreased, as you can see in the red color, also in the quantification below. So 6-mercaptopurine is working both on the right ventricle side. 6-mercaptopurine also working on the lung side. It seems very beneficial. We tested this in the animal model. We also give 6-mercaptopurine as a prevention strategy. That means we give 6-mercaptopurine before inducing the disease in the rats, and it's much more stronger than this treatment strategy. We didn't leave just like many other publications, just oh, this drug works and that drug works. We actually used 6-mercaptopurine to test in the patient. We didn't leave just like many other publications, just oh, this drug works. Patient 6-mercaptopurine. This study just been published only 1 month ago in the highest impact factor of the respiratory field. So in this, I won't discuss the details of this publication. What 6-mercaptopurine is doing, it's actually increasing the same benefits that we have seen in the rats. So the finding that we see in the in vitro cell culture, the findings that we seen in the animals can be translated to the patients. However, there are some substantial side effects that has to be improved, because 6-mercaptopurine we gave as a drinking water or by orally as a tablets to the patients, and what it seems like there is some side effects like neutropenia and some other side effects like thrombocytopenia are observed in these patients. Of course, since patients are dying, it's important to have some medication having compared to having no medication. So therefore, we continuing this treatment for the time being in these patients. And I think my computer screen. 1 second, please. So since 6-mercaptopurine is working in the patients, we are aiming to develop a more specific targets of NUR77, such as by taking 6-mercaptopurine as a template. So we are using docking approaches and also other approaches to make a more stable and more efficient formulation that can actually increase the neurosemestine expression in these patients, because we have very strong evidence that 6-micropirin is working in almost in the animals and then also in the patients. And the last, I'm going to discuss about a project called RVX2A drug that we developed. And the RVX2A drug targets in vivo a protein called BRD4. What is BRD4? BRD4 is a protein. It's a gene actually releases when there is a DNA damage. So in the patients of PH, there is a DNA damage going on. So when there is a DNA damage response in these patients, the BRD4 expression is substantially being increased. The BRD4 DNA damage leads to, if DNA damage is continuing in this lung tissue, there is increased proliferation of the cell, there is inflammation, and there is something else is going on. So this we have a very strong evidence, and therefore we think BRD4 has a very important role in PH. So BRD4 works on the DNA level. It can actually, it's a epigenetic switch. And in these patients, we have a DNA damage, and in these PH patients, the BRD4 expression is substantially being increased. And this has been confirmed by our collaborators independently without knowing that we are working on the same project in Canada and also in Germany. So therefore, since the evidence is very strong, we tested we developed a drug called RVX208. This RVX208 is specifically targets BRD4, not even a other family member of the same family. So BRD4 as RVX208 is very high specificity for BRD4. And as you can see in this top panel, we're looking at cell proliferation by looking at Ki67 cells. The blue stands for nuclei of the cell, and the pink color stands for the proliferative cells. The more pink color means more cells proliferating in the vehicle group. But if you give RVX208 on the top right, you can see the proliferation is being decreased. And at the same time, while reducing this RVX proliferation of the cells, it also increases the apoptosis, which can be seen by the staining of annexin. On the right side, RVX208 can increase annexin 5 staining. That means RVX208 is reducing proliferation, increasing apoptosis of cells in this patient. And at the same time, what can be seen by the staining of annexin 5, increasing apoptosis of cells in this patient we have are actually isolated from the patients who either been transplanted with the lung transplantation or died. So RVX208 also doing much more things than what we expected, which actually does decreasing the inflammation, for example. It also maintaining a balance between these TGP-beta BMP signaling pathways, which are changed, like I discussed before. So RVX208, I'm not going to the details, but what you can see, RVX208 is involved in almost all the pathological features of the project called RVX208. What is BRD4? Reducing inflammation. BRD4 expression is substantially is continuing in this lung, have very strong evidence, and it can actually, and it is blocking cell cycle in Canada and also in and so on. And the last, we tested this, and the RVX2 tested in Canada before knowing that they were working on the same project. And we also tested in RVX2 in this, and as you can see in this slide on the top panel, you can see the pH development. The animals are treated with RVX can be rescued. So all these black dots actually are vessels in the lungs of these rats. So the same phenomenon also happened in the patients. So this black dots means the vessels are completely occluded. The blood cannot be taken up by these lungs. So, but in if you give RVX, as you can appreciate that almost all the vessels are opened up just by giving the drug only for 14 days by drinking water. So we didn't even give the drug by lock. So RVX208 also maintaining a balance between to the lungs by aerosols and so on. We gave the almost all the pathological, and it's also blocking. We tested this RVX208 lungs of these rats. And a very substantial contribution to the field of the pH so far in the history of the pH. So we are the first to publish a first multi-center preclinical trial in pH research, and this got a lot of attention across the globe in different countries. and this study was just published end of last year. And this we're really proud to say that this is publication which got the highest attention in the last 6 months in the respiratory field across all the different diseases. So it has been cited very well, and it has been commented at least 6 times by different groups across the globe positively, saying that this could be the possible treatment. So this is an ongoing study, and what we are doing now is next to where we are bringing to the clinical trial. We in collaboration with our Canadian group, also in Amsterdam, just to say that our wish to wait when we said to the patients and to the government that we want to do a trial, and we got 3.2 million euros in one day. So that shows that patients believed in us, the government believed in us, and we got money raised in less than 6 hours, 3.2 million euros in just to support this clinical trial. And we think the RVX2 might make a big change. And this trial is registered now, and it's ongoing now, and the initial results are very promising already. So what I've been trying to show you using very superficial data, because I couldn't show you all the data. We used, like I mentioned, we used different approaches. We used combined different disciplines. We use chemistry, we use clinical medicine, we use patient samples, we use chemistry, we also use some pharmacology, so what we did is what I tried to show you. We use BMP9. We use BMP9 to increase BMP expression. We used RVX to increase BMP expression. We use 6MP, 6-methylpurine to increase neurotransmitter expression, thereby increasing BMP expression. Stop disease progression. And RVX is also working at different level. And we are also showing FK506. Currently, we are waiting for the funding to this clinical trial also feasible. With me and in collaboration with our research groups in Canada, also in Stanford in US, and in Amsterdam. So this is basically the summary of my presentation. Of course, I didn't do this myself. I have several clinical collaborators, basically professors, and I obtained different funding. And recently, I've been awarded with 1.1 million euros from different pharma companies and also from the European Union to continue my funding research and to understand and to explore what's going around in these patients, especially in the female patients, why they actually developing this disease much faster than the male. And I would like to thank all the people, and thank you for your attention. And I hope some things are clear for you. And I'm happy to take any questions if you have.
Thank you, Dr. Panababu. To deliver webinar from molecular level, I feel it's a very knowledge imparting and enriching session. Now, I ask our participants to clarify their doubts one by one by raising their hand, please.
Okay, thank you, Dr. Panababu, to deliver this webinar from molecular level. I feel it's a very knowledge imparting and enriching session. Now I ask our participants to clarify their doubts one by one by raising their hand. Please, Mr. Kamesh, Assistant Professor, to take care about the same. Please, if anybody raised their hand, please allow them to ask their questionnaire.
Okay, sir. Thank you, sir. Thank you for giving valuable information about pulmonary hypertension. Now coming to questionnaire section, first one you will giving to Kinnu. Hello.
Hello.
Hello. Okay, sir.
Please unmute him. Please unmute that person. Somebody is raising their hand showing number 4.
Excuse me, sir. I will ask the questions of Kinnu. Is there a loss of bone morphogenetic protein type 2 receptor potentiate the effects of bone morphogenetic protein?
Yeah, that's true. So the BMP ligands which are present in our circulation of every human individual can actually increase the BMPA2 expression. So the BMPA2 expression leads to, of course, reversing this disease process. But what I was mentioning that BMP ligands also can induce calcification in different parts of the body if you give it. Is it answer to the question?
Yes, sir. Next, another more questions from Charan.
If you unable to ask, just type down. We
Hello, sir.
will ask.
Okay, Allah, sir. Another question.
I typed, sir.
Okay, sir. Okay, sir.
Okay, carry on.
Carry on.
Sir, hello, sir.
Oh, yes, sir.
Sir, is taking more salt lead to the hypertension, sir?
Sorry, can you
He's
repeat again, please?
He's taking more salt, sir. Sodium chloride leads to the hypertension, sir. High BP.
Yeah, so taking more sodium chloride, of course, for prolonged time, of course, can lead to hypertension. But in the but we haven't observed at least in the rats for giving for 14 days. So perhaps it might take much more time, and also about the concentration of the sodium chloride is important, of course. How much sodium chloride is actually being placed.
Okay, sir.
Thank you.
Thank you, sir.
Giving for 14
Pawan,
days for the
sir.
concentration of the sodium.
Pawan. Next, another one is Mahata Priyanka. Allow to talk.
Hello.
Hello. Mahata Priyanka.
Hello.
Okay, okay.
So
Pawan.
I just have a question.
Pawan Kumar.
Okay, I just have two questions. One, have you looked at the endothelial protection agents in the PHS model, PAH model, like C1 inhibitor or heparin, warfarin? Does it at all any effect on the PAH in terms of remodeling while remodeling endothelium dysfunction?
I didn't get the last part of the question. Sorry.
Okay, actually the hallmark of in this PAH is the vascular remodeling. So that means endothelial dysfunction. So have you looked at the protecting the endothelium in the first place?
So these drugs actually works on the endothelium production. So they actually maintain the signaling. They repair the endothelial dysfunction, which I didn't talk. They repair the endothelial dysfunction by targeting BMP2.
So I was wondering, can we target this TGF pathway using the way they use in the cancer, in treating cancer, like using anti-TGF beta antibody along with BMP9 administration to the patient, so that I know maybe it could have better results, I guess.
So antibody for what? Hello? Antibody for what?
Sorry, sir.
I understand that co-administration is the BMP9, but antibody for, yeah? I missed what is the antibody co-administration with the antibody. I don't know what the speaker is asking. The participant is asking.
Pavan, are you there?
Okay, no sir, no.
Giving BMP.
Pavan, are you there?
No sir.
Yeah, anti-TGF antibody. He is asking. Just he typed.
Okay. Got it. Yeah. So it's possible that giving anti-TGF antibody can rescue this phenotype. So it's also under clinical trial with our collaborators and in the Harvard. So our collaborator is giving anti-TGF antibody. He could reverse it, but the problem is also inducing some other off-target effects. So it's actually trying to develop with a company a much more specific one. Despite its because TGF-beta is important for almost all the cellular process in the body. So giving antibody, taking out TGF-beta may be not a good idea because it's important for normal living as well for different processes. So they're actually titrating the different concentrations of the antibody to make sure they don't take everything, but at the same time they take enough to rescue this phenotype. Yeah, there is one other trial is already just approved, giving a different kind of antibody can actually block this same pathway. So it's going on. So there are some people interested in the monoclonal antibodies against changing this signaling pathways. Yeah, we are primarily interested in pharmacological approach in combination with, of course, the molecular biology and clinical medicine. Yeah.
Any questions, Kamesh?
Yes sir.
Okay, please ask.
Another question from Priyanka. She is not in group. This question is ADR effects of 6-mercaptopurine in pulmonary arterial hypertension patients.
Adverse drug reactions of mercaptopurine.
Okay. Yeah, the 6-mercaptopurine has several side effects, like I mentioned. So they have the patient's experience. We give the 6-mercaptopurine for 16 weeks to the patient orally by as a tablets, and those concentration, the doses that we gave to the patients is the same doses that they gave it to the inflammatory bowel patients, for example. Despite giving the same doses, we in this patient, PH patients, we experienced several side effects. The most commonly was neutropenia and thrombocytopenia. It's the biggest problem. And 6-mercaptopurine is also an immunosuppressive drug. So it has been used in the organ transplantation over 60 years as an additive immunosuppressant for to avoid the rejection of the transplants. So despite giving the same doses, seems like our patients, our PH patients are very sensitive. So they're experiencing much more serious side effects than the normal other patients, like inflammatory bowel patients. So what we are trying to achieve is now make this 6-mercaptopurine a specific delivery to the lungs, or targeting neurosemensism different agonists to make them much stronger. But at least this pathway seems working.
Okay. And another question from Pavan, that any KO model for BMP9?
Yeah, so there are different knockout models available for the BMP9, and they have some conflicting data. So just been published earlier this year, only 3 months ago. The BMP9 knockout models, depending on the place where they studied and depending on the model they studied, they have different phenotype. So in one study by France group, our collaborator actually shows that BMP9 may not be working. They actually promoting, and the other model shows opposite. So I think in this respect, not taking out BMP9 to understand this phenomenon may be not a good idea, because BMP9 is very important for normal cells as well. Whereas the problem in PH patients lies in the lungs only. So it's important to understand taking out the BMP9 in a lung-specific BMP9 knockout model, rather than taking out BMP9 globally. So there are much more studies going on. Like I said, the UK group, our collaborator, and at the Imperial College London, they actually developing BMP10 as a therapeutic target and they are testing in the now in the animal models, which seems very promising. I just saw the data last week, and the company is actually moving forward to test on the patients now in the UK patients. Yeah.
Thank you. Any questions are there, Kamesh?
Yes, sir. Asking from Pravan.
Just allow only one person to ask the question.
Okay, sir. Okay, sir. Last question. At endothelial production agents in pulmonary arterial hypertension.
Yeah.
It is not clear. Can you ask you again?
Sir. At endothelial production agents in pulmonary arterial hypertension.
Yeah, the problem in these endothelial cells from these patients are dysfunctional. So they're not working in the way they should. So they actually being dysfunctional. For example, endothelial cells should be touching to each other, for example. So what is happening now is they have a leaky function. For example, these endothelial cells are leaky. So the other blood cells, like immune cells, are going inside the lung, blood vessel. They're also inducing inflammation. They have apoptosis problem. They have proliferation problem. Basically, they have almost all the problem that we see in the cancer. So there is no endothelial production happening in the patients. That's why we want to have endothelial production. And the current treatment that we have in the clinic now to reduce the symptoms are actually trying to work on endothelial cells as well. For example, vasodilators are being given to the patients now at this moment. They're trying to rescue endothelial dysfunction, but they're not successful. So there is no endothelial production so far. So what our aim is to look at the endothelial dysfunction very carefully, and we believe that endothelial dysfunction is the main trigger which is starting the disease. So that's why we develop these drugs only using patient endothelial cells. So all these drugs, what I told you today, is already improving the endothelial function. They're actually endothelial protective. We did also electron microscopy to see how these endothelial cells are behaving in vivo, to see the junctions and so on. These endothelial cells are becoming normal after giving this drug for at least for 2 weeks.
Thank you, Babu. And we have another question from Satya Prasad. Please allow him.
Okay, sir.
Dr. Satya Prasad.
Sir, good afternoon, sir.
Yeah,
Sir, this is Dr. Satya from Koringa College of Pharmacy. Sir, I have one question for related to the pulmonary arterial hypertension, sir. Sir, my question is, how to affect the sitaxanthin in related to the pulmonary arterial hypertension, sir?
Can you repeat the first part?
Effect of sitaxanthin, sir. Sitaxanthin drug.
Okay, I don't know about that one, but yeah. So how to assess? You mean that's the question?
Okay, sir.
So yeah, of course, our approach is completely depends, like I mentioned earlier. So our approach is completely starts from the patient perspective. So if your drug has an effect on this, one of the signaling pathways has been changing in the cells of this patient, yeah, definitely you can trust your drug in this cells. And of course, collaboration is possible to obtain these patient cells.
Okay, sir. Hello, sir.
Yeah, go ahead.
I have one more question, sir. What is the hemodynamic functions for pulmonary arterial hypertension, sir? Hemodynamic functions. Actually, I'm a pharmaceutics person. So I have some doubts for my research work related to the pulmonary arterial hypertension drugs and development. Sir, what is hemodynamic function?
Yeah. So the hemodynamics basically is a broad term. So what's being used in it is being used in the clinic to diagnose the patients. For example, if I have PH, I go to the clinic and the pulmonologist or cardiologist actually determine my hemodynamics. What he does is you look for the like you look at the blood pressure. Instead of looking for the blood pressure, they look for the pulmonary artery pressure, because the problem here in this case is not in the other parts of the body. like in the left ventricle disease. So
Okay.
what they do, they look at the lung pressure. So the blood has to flow from the right ventricle to the lungs, of course. So the pressure in the pulmonary artery is being changed. The pulmonary artery pressure is being increased in this patient. So those pressures will be determined in the clinic, in the hospital, by either by pulmonologist or a cardiologist. And they say, oh, if the pulmonary artery pressure is 25 millimeter mercury, they can say, oh, this guy has a pulmonary arterial hypertension, and they start treatment.
Okay, sir. Okay, sir. Okay, thank you, sir. Thank you for valuable information. Thank you, sir.
You're welcome.
Kanakaraj garu?
Sir.
Yeah, sir. Please carry on, sir.
Good afternoon, everybody. Sir Kondababu garu, thank you very much to accept our invitation, come for international web seminar. We are give valuable information to the who participated in the seminar. So 1200 registration, it is a miracle. We are thankful to the Pani sir and their team work who are behind the Pani sir, that is Kamesh, Kumar, Prem, and other staff who are well performed for the 1200 registration. Kondababu garu, we are very thankful, and again we will utilize your service whenever you need your services. We will welcome you, sir. We are again thank on behalf of the Koringa and other institutions for giving a wonderful information through this webinar, sir. Thank you, Kondababu garu. Thank you.
Thank you, Kanakaraj garu. Thank you, sir.
Thanks a lot.
Thank you, sir. I ask Secretary Madam to speak some words regarding the session.
Madam is in YouTube.
Okay, sir. Okay, sir. Okay, sir. Thank you. Thank you, Kondababu garu, for your interest towards our college and accepting our invitation. And I thank who supported me a lot for this webinar session. I want to say their names too, because it is a good achievement to register such a big number. Namely, I wanted to read out their names. Dr. Bhaskar Raj garu, Vasvi College, Dr. Mohan Gandhi, Vasvi College, Dr. Nageswarrao garu, Director, KJR College, Dr. Ajay and Dr. Raghavendra, Ma'am College, Dr. Pavan, Riper Institution, Dr. Mishra garu, Emmanuel College, Dr. Chakradhar, Emmanuel College, Dr. A. Lakshman Rao garu, VV Institute of Pharmaceutical Sciences, Dr. Jagadish Panda garu, Raghu Institute of Pharmaceutical Sciences, and Dr. Ashok Tulluru, Vishnu College of Pharmacy. And I especially thank Dr. Bhavani, Koringa College of Pharmacy Principal, Dr. Satya Prasad, Koringa College of Pharmacy, and Mr. Vasbabu, Faculty, Vikas College of Pharmaceutical Sciences, Mr. Girish, 4th B Farm, Vikas Institute of Pharmaceutical Sciences, Mr. Prudvi, finally M-Farm, Vikas Institute of Pharmaceutical Sciences, and finally, thank my management of Adarsh College of Pharmacy, Koringa College of Pharmacy to provide a great chance to conduct this international webinar. If I missed out any names who helped me a lot, I'm very sorry about the same. And because you have shown a great consent towards my institution as well as me, and I love you all. Now I ask to speak any words regarding the session. Dr. Kondababu garu. I think he left, sir. Dr. Kondababu garu. Dr. Kondababu garu. Yes, sir. Now I ask our sister concern institution principal, Koringa College of Pharmacy, Dr. Bhavani Garu to speak about the session as well as next proceedings too.
Okay, thank you. Thank you, Funny.
Thank you, madam.
Yeah. Sir Kondababu, sir, you have given a tremendous and very useful lecture, especially targeting to the pulmonary drug delivery system, and which is very innovative thing. I hope much of the work is going on there. Hope for the next in future, India also going to be developed, that academics also we are going to develop like that modules and models. Thank you, sir, for your presence in this occasion. Thank you very much. I hope all the participants have enjoyed the session by our sir, and thank you, Funny. You have invited very nice personality for presenting the good thing.
Thank you, madam.
Yeah, and I hope all the participants till now you know that we have tomorrow onwards we are inaugurating and we are conducting a international webinar session, especially Arogya Rakshak Pharma Series 2, that is interaction with the expert pool. So I hope everyone do participate in that, and whatever the queries, eminent personalities are coming. So this is a good platform where you can ask a number of questions. Whatever the doubts you have, you can clarify with the leaders, means who are the top in their professional field. Thank you. Thank you.
Thank you, Bhavani, madam. Sir Kanagaraj garu, I invite you to speak again, please.
Thank you. Thank you to the Pandigaru and his team. Sujini madam, Saujanya, Bhaskar garu, Prem, and Kamesh, Sivaram Krishna, Srinivasa, and the other staff, Venkateswar and all who are with you. It is a good teamwork. It is a definitely wonderful session given by the Kondababu garu. I congratulate Pandigaru and his team. In future, we will going to conduct so many international seminars. It very help to you. Very good. Congratulations to all. Thank you to all the participants who are attending this webinar. Thank you.
Thank you, sir. And coming to the feedback forms, guys, we will send you through the email ID, which one you have given in the registration form. So you just check those your email ID and fill the feedback forms. Then you will get the certificate. See, there are 1200 members as participants. That's why we can send you only 100 feedback forms per day. That's why it may take 2 weeks. So don't worry about your certificate. We will take care about the same. If you have any doubts regarding the certificate, you can directly call us. We have given our contact number on the brochure. You can take out and you can call us at any time. Okay, definitely we will respond. We have given 2 mobile numbers as a contact numbers, so you can use those numbers to contact us. Okay, thank you very much. Thank you all for participating in the same, and I ask you to participate in the next seminar, I mean webinar. So Bhavani Garu has invited leaders throughout this India. So it may be very useful to you. Whenever we are having, they are availing great resource persons to us, definitely we have to utilize that. It is my opinion. So as a co-professional, I can suggest you that definitely you have to take their, what's that feed, definitely, to gain the knowledge or to enrich the knowledge. Thank you, all of you. Thank you, Kondababu Garu. Kondababu Garu, you left before, so I ask you to speak about this session, a very few words. Okay, thank you. Sir, please speak out, sir. Kondababu Garu, Dr. Kondababu Garu.
Sorry, can you repeat the last one?
Sir,
the voice is broken.
Sir, just you speak about this webinar, sir. Hello? Is it not audible to you?
Yeah, so it's a pleasant to be invited. Thank you, Dr. Kanu, Dr. Kanakaru, and others for inviting me, and I'm really really enjoyed, but of course I will combine with expertise. I know that I tried my best to keep it very low, but I understand that many of the students are from the pharmacy, but in this case, my presentation is involving the voice is broken disciplines, but it is just to show that many disciplines, but it is also process.
It is not audible, sir.
Yeah, is it audible to you? Is it audible to you now?
Yes, sir.
Okay, okay, Dr. Kondababu garu. Yeah, okay, Dr. Kondababu garu, to participate in this webinar. Thank you very much. This is all about this session. Just let us sign off. Thank you all, and let us meet again on 11th with another webinar. Thank you very much.
Thank you. Thank you all.
Thank you all.
Thank you, sir.
Give a flavor of
Ramesh.